Hi, this is Sajida Kilani, and this is an explanation and a review of the drug-receptor interactions and pharmacodynamics. It is Chapter 2 from Lippincot's Illustrated Review, the textbook of pharmacology. First of all, what does pharmacodynamics mean? Pharmacodynamics is concerned with two main ideas. The first one is the exact effect of a certain drug on the body. Like when you give a certain drug, what this drug is going to do on this body. The other thing is the effect of the concentration of this drug on the magnitude of the response that we are going to get in this body. Like if you increase the concentration or decrease the concentration, what is the response? How does this affect the response of this body from this drug? Okay, whether you are going to get a better response, you are going to get a bad effect. So the effect of the concentration. Now, the effect of the drug on the body could be one of two. It could be beneficial, like when you give a certain drug and you get the response that you are looking for, that you have given the drug for in the first place. And when we get a beneficial effect, this is the therapeutic effect, العلاجي. And we could have a harmful effect. This harmful effect is additional effect we are not looking for. This is what we call the side effect of the drug. Okay, now in this chapter we're going to explain how do we finally get the beneficial or the harmful effect of the drug. Now this is a drug and you've taken this drug, and from chapter 1 you know how the pharmacokinetics will affect the drug until you have these final effective substances or molecules of this drug. Now, after reaching this substance, we should get an effect. This effect of the drug is there by two ways, okay? The first one is what we call the others category. It contains many possible ways, and I will take just one example of it. It is, for example, the chemical interactions. And the other way is what we are concerned with in this chapter. So, the chemical interaction. For example, you are suffering from a heartburn in your stomach. You have excess acidity in your stomach. How can we fight this acidity and treat it by drugs? Simply, you take a drug that is basic in nature, and this base will interact with this acid. It is gonna fight the acidity, and heartburn is over. Now the other way is what we call the drug-receptor complex. Now, the drug-receptor complex means that this drug that we have given should go inside the body and find a receptor and form a complex between with this receptor. And when we have this complex, we are going to get this effect. Like for example, let's take one of these. This is the drug molecule, the ultimate drug molecule. This drug molecule is going to work as a signal that has a certain message. It is carrying a certain message inside it, and this message should be sent and given to a certain protein molecule that is the receptor. So if this signal is circulating inside your body and it has reached a certain cell here, this is the lipid bilayer of a certain cell, this is the extracellular space, and this is the intracellular space, and this signal should find a certain protein molecule. It is either on the cell surface or inside the cell, where this signal should cross to the inside of the cell, and then it should bind to it. When it is bound to it, this complex will be activated. It's like putting the magical key inside a certain lock of a certain door, and when they are together, there will be a response. Okay, it is activated and there will be a response in this cell. So ultimately we will get a certain effect, a certain response. Okay, now you remember from biochemistry the lock and key theory? This signal, this drug, okay, here is like the key, and this protein molecule, this is the receptor, is the lock. Now there should be specificity. Okay, these are specific. This is specifically synthesized or made or there to be fitting in this receptor and this lock specifically. This is one. The other thing is this receptor, this drug, this key, will either bind to a protein molecule on the cell surface or it will bind to a protein molecule inside the cell. Keep in mind that this part here is a lipid bilayer, and that this here outside the cell and inside the cell is water-based, is a hydrophilic environment. Okay, now the third thing is that when this signal, this key, is bound to this receptor, there is the complex, we've got the complex, there should always be a response, an effect, a change. Okay, so the cell before and after the binding is never the same. There should be always a response or an effect or a change on this cell. Now let's get back to this stomach that we've drawn here, and let's imagine that this is the layer of the cells covering the stomach. Let's take one of these. Okay, this is here the cell, this is the lipid bilayer, and this cell here is working to secrete acid, the H+. Now if we are going to treat the heartburn in a way of the drug receptor complex, there should be a protein molecule here or here in the inside of the cell, and the drug should come and bind specifically to this protein molecule, to this receptor, and when they are bound, we will make a certain response, a certain effect, a certain change, that this change will prevent the cell of synthesizing or secreting acid anymore. So less acidity inside the stomach and we are fine. Okay, so this is an example of how drugs can work in two different ways. The one that is simple, very simple chemical interaction, and the other way is the drug receptor complex that is more complicated and that works in a different way. Okay, now the very last idea that I want to explain here is that when you have, for example, this person, our friend here, has an abscess in his finger, for example, okay, and there is inside this abscess bacteria. This is the bacterial cell here. Okay. Now, he decided to take an antibiotic, okay? And this antibiotic should work on the bacteria in the abscess of the finger in his left hand. How does this drug know that it should go to this specific site? Actually, it does not. But what happens is that this drug that you have taken, and after all the pharmacokinetics, the absorption, the metabolism, the distribution, it is circulating inside the body. It will go to every specific part of the body, to every specific site that it can reach with the blood. So it will reach to the hand, it will reach to the nose, it will reach to the stomach, it will reach to everywhere, but it knows that it will only work on a specific receptor that it is made for, its target cell, its target receptor. So when we say this is the target cell, because it has the certain receptor. So the target cell is the bacteria, this is the target cell, this is the specific receptor that this drug here, the antibiotic, can work on, because this is where it fits, okay? And so it will work on this bacteria and everything is fine. So it does not know that it has to go to the finger only. It will circulate in all the body. And for example, if there was a cell inside the body that has a similar receptor to the receptor that's found on the bacterial cell, it will go and it will interact with it. And this is why we sometimes have side effects of certain drugs, okay? Because the therapeutic effect that we are concerned with here is to kill the bacteria, because we have this receptor here. But the side effects and the harmful effect, for example, we might get because this drug has found another receptor similar on a cell in the body of this person, okay? And the last thing you should know is that this signal here, this key, this part, this molecule of the drug that is going to bind ultimately to this receptor is what we call the ligand, okay? So when we say a ligand, we mean the drug, but the ultimate part, the ultimate molecule of this drug. Because sometimes we might give additional modifications, additional substances, we add them to this drug, for example, to avoid the acidity of the stomach, or for example, to make it more absorbable. But what we are concerned with here is the very pure molecule that is going to make the specific effect of this drug. Now let's take one of the receptors in here. Now what a receptor is, it is a protein, a certain protein, polypeptide, with certain three-dimensionals and three-dimensional conformation. And this protein, this receptor, has certain sites that are available for certain ligands to come and bind to them. Receptors might have one or more than an active or a binding site for the ligands, okay? Now, they used to think that this receptor, when it is just there sitting on the cell and without any ligand bound, it will be inactive, okay? This is the symbol of a capital R. And when a certain ligand comes and binds and activates this receptor, so the receptor will be active. This is the R with a star. Now what they have recently found, that if we have, okay, this cell surface with 1, 2, 3, for example, receptors, okay, this is the same receptor. These are of the same type of receptor, the same family. And these three brothers are not bound to any ligand. They found that we will find a certain percentile, okay, a certain percentage, of these receptors will be active even without being bound to a ligand. So, for example, this is inactive, this is inactive, this is the R, this is the R, this is the R with a star, okay? Now, this is in the absence of any ligand. So, there is a baseline activity of these receptors, okay? So, there is, how does this work? There is an equilibrium, okay? And this equilibrium is between the R and the R star, the active and the inactive state, but it favors the inactive state, okay? So these receptors will be in an equilibrium of the inactive and the active state, but they are favoring the inactive, but we are still having an active, some sort of activity in the baseline in the absence of any ligand. So the activity of the receptors in the absence of the ligands, it is never zero, okay? It is never zero, it is always higher than zero. Now, receptors in general are divided into two main categories: the intracellular receptors and the cell surface receptors, okay? Now, let's begin with the intracellular receptors. Let's come back to what we said in here. Now, we said that the signal or the ligand will come and bind to a receptor, whether on the cell surface or in the inside of the cell. Now, what controls this is, as you remember, we said also that the extra and the intracellular space is a hydrophilic environment, while this here, the lipid bilayer, is hydrophobic environment. Now, in order for this ligand to cross to the inside of the cell to go and attach and activate the intracellular receptor, there is here a barrier. This barrier here is the lipid bilayer membrane, so it is a hydrophobic membrane, okay? Now, this was the same diagram, and this is the lipid bilayer. Now, in order for the ligand to cross this lipid bilayer, it should have some sort of lipid solubility. So, when the receptor is intracellular, its ligand should be lipid soluble. So, it comes and finds the receptor in here, and it will bind to the receptor, and everything is fine. Now, the problem here is that the extracellular space here, the blood, the extracellular space, everything in here is based on water, so it is hydrophilic. It should be water soluble, okay? Things should be water soluble to be able to move freely in here. So for this drug, for this ligand to reach the intracellular receptor, it should have a vehicle, a carrier, a transporter in the blood and the extracellular space until it reaches here. So there should be this carrier here. This carrier will also take it through the extracellular space until it reaches to the lipid bilayer, to the cell membrane, and they both dissociate, okay? they will dissociate. The ligand only will come and cross and reaches the intracellular space. Now back again, the same problem is here. We have a water environment inside. So there should be also a carrier, and this carrier will also take the ligand until it reaches to the receptor, dissociate, and this is activated. Okay, now usually this carrier here is the albumin. Okay, the protein albumin. This is the first thing. Now the other thing here is that the receptor of the intracellular receptor is usually growth factors, ribosomes, okay, and things that are found in the inside of the cells, transcription factors, sorry, or ribosomes. Now, if this ligand comes and binds to a certain transcription factor in the inside of the cell, now what we have inside the cell here is the nucleus. Okay, now in the inside of the cell we have the nucleus and we have the DNA here. These two will go to the inside after this has been activated, and they will go and bind to a certain site in the DNA or to a certain site on the RNA, and they will affect the transcription, translation, protein synthesis of the cell. So we will result finally with a certain protein, for example, or they will come and affect ribosomes. They bind to ribosomes, the receptors are ribosomes, activate the ribosomes, and affect the translation of the RNA to a protein. So we'll result with protein, with polypeptides. Okay? And the last thing that you should know here is that this whole process of reaching, of being carried, of reaching the nucleus, of transcription, translation, protein synthesis, all these things, they will take a long time. So the effect of these drugs is usually hours to days. So we don't get an immediate effect, an immediate response of the drug on the body. We don't see it immediately. Now the other type of receptors is the cell surface receptors. So this is the cell surface, and we will have the receptor here embedded within the lipid bilayer. Okay, now if this was the lipid bilayer, we still have this watery environment, so the drug can simply just move and reach the cell surface. It does not have to cross the lipid bilayer, so it can just come and bind here. So the ligands usually are, or they are hydrophilic. Okay, now in this family, or in this category, we have three families. Okay, the first one and the simplest one is the ligand-gated ion channels. What do we mean by this? Now, this is, okay, it's clear. Now, if this was the cell membrane here, we will have here a certain channel. It's a door, okay? Certain gate. Now, this gate here has a certain site. This protein is a gate, and this gate has a certain site for the binding, as we said before. And here, the ligand will come and bind. When it binds, this will be activated. When it is activated, there will be conformational changes at the level of the three-dimensional or the four-dimensional, mostly the three-dimensional level of the structure of this protein. And now this gate is open. Now, usually each gate is specific, each protein is specific for a certain ion. Like for example, let's assume this was a sodium channel. Now, if these Na molecules, the sodium molecules, are trying to pass here, it is closed, so it will not be able to cross. Now, when this ligand is bound and the channel is active, we can easily now make the sodium cross to the inside of the cell. So the gate is open and sodium will cross to the inside. So now we have Na in the intracellular space. Okay. Now, as we said, usually each gate is specific for a specific ion. Like for example, we have these gates here. Okay. And this gate here is now activated by the binding of the ligand, and it is now open. This specific gate is there for the passage of chloride ion. Now, the effect that you're going to get from making the sodium, the positive ions, get inside the cell is different from the effect that you're going to get by making the chloride, the negative ions, get inside the cell. How is that? Let's go back to the physiology of the cell, to the physiology of the action potential. Now remember that the cell at rest is in the negative voltage. Okay, if you are taking the inside of the cell, the voltage, the charge of the inside of the cell, it is usually negative. Now when you get positive ions inside the cell, this is going to get higher until you reach a certain thing that we call the threshold. Okay, now this is the entrance of the positive ions to the inside of the cell. We reach the threshold, we're going to reach the action potential level in here. This is the positive that we are looking for. Now when you reach this, this is called the depolarization phase. Okay, then we're going to get down back to the negative level. Okay, now the negative ions will get inside or the positive ions will go outside. This is the repolarization. Now you get down until you are below the resting potential, and now we are in the hyperpolarization phase. This is by getting more negative ions inside or by taking more positive ions to the outside, usually by getting more negative ions. Now we are back to the resting potential here until another action potential comes. Okay, so what does this has to do with that is that the nicotinic receptors that are the sodium, okay, the real sodium channels inside your body, okay, they are activated by this ligand here, which is the acetylcholine. Okay, when acetylcholine comes and binds to the nicotinic receptors, the channel will be opened. When this channel is open, the Na, the sodium, will get to the inside. Now we are depolarizing the cell, so we are activating the cell. This is usually why the nicotinic receptors are activatory. Okay, they are activating the cell. While on the other hand, the realistic example of the chloride channels inside your body, especially in your brain, we have the GABA channels. The GABA channels are ion channels for the chloride channels. Okay. These channels here, the ligand for them is the benzodiazepine. Okay, it is a sort of drug. So this benzodiazepine, BZD here, okay, will come and bind to the GABA channels, so chloride will get inside. Now when chloride is getting inside, we are either repolarizing the cell, or it is in the resting potential and we are hyperpolarizing the cell. Now if this cell is at the resting potential and we activated the GABA channels, chloride is inside, we are hyperpolarizing the cell. So for the action potential to come and appear in this cell, we need more and higher Na influx or positive ions influx. So this is inhibitory. Okay, so the GABA channels are usually inhibitory. This is the first type of the receptors, the cell surface receptors. They are the ligand-gated ion channels. They allow certain ions to get inside by binding to a certain, the ligand should bind to a certain site in the channel. This is the key, okay, the ligand and the lock. It's like opening a door simply, okay? Now the response here will take only milliseconds, okay? It is very fast because you just open the channel, or you just open the channel and the ions will get to the inside. In comparison with this intracellular, he said they will take hours to days. Just to make things clear, this is one. Now the other type of the cell surface receptors is the enzyme-linked receptors. Okay, this is number 2. Now the enzyme-linked receptor, the receptor on the cell surface here, okay, is a protein molecule in a certain conformational structure. Now the ligand comes and binds to this receptor. The receptor has two main parts: the extracellular domain and the intracellular domain. Okay, this is the lipid bilayer. Now in the extracellular domain, we have this site that the ligand will come and bind to. In the intracellular domain, we have a certain enzyme here. This enzyme is usually inactive. In the inactive state of the receptor, this enzyme is inactive. When the ligand comes and binds to this receptor here, it will activate the enzyme here. This is one. When this enzyme is activated, this enzyme is going to work on a certain substance in the inside of the cell and make it active. For example, let's take a realistic example of this in the inside of your body. This ligand comes and binds to this receptor. Now the most common and the broadest one is that the cell inside the body, is the tyrosine kinase linked receptors inside the body. The tyrosine kinase is the enzyme in here. When you phosphorylate the tyrosine kinase, it is going to be active. Now the activity or the job of this tyrosine kinase is to get to a certain enzyme, a certain protein here, and it will lead to the activation of this enzyme here or this protein here by phosphorylating it. So after it has been autophosphorylated, it will phosphorylate another molecule in here, and this molecule will go and elect a certain effect in the inside of the cell, work on certain thing. Okay, now this molecule in here is what we usually call, this one here is what we usually call a second messenger. Second messenger means that there is a messenger, okay, that takes the signal from the first one here, which is the first messenger is the ligand, and carries a certain message, a certain signal. This signal will be transmitted and it will reach the second messenger here, and the second messenger here will take the ride, and it will go and make the certain effect. Okay. Now, example of this is insulin. Okay, insulin, the ligand here, has an insulin receptor on the cell surface. Okay, now the insulin. Now, a realistic important example of this is the insulin. If this ligand was insulin, and this was the insulin receptor in here, okay, upon binding of the insulin to the insulin receptor on the cell surface, it will make certain conformational changes that will autophosphorylate the enzyme here, okay, the tyrosine kinase linked to the insulin receptor, and this will make activation and phosphorylation of certain second messengers, certain molecules in here. In certain cascades, this will lead to the glucose intake, to increase the glucose intake in the inside of this cell. Okay, other examples include the PDGF, platelet-derived growth factor, and the EGF, the epidermal growth factor. Okay, now the effect of this enzyme-linked receptors binding of the ligand, it needs minutes to hours. Okay, so it has faster effect than the intracellular, but longer time, needs longer time than the ligand-gated ion channels. Okay, now the last and the third example of the cell surface receptors is a very important one. It might be the most important one, and it is the I think it is the broadest. Okay, and it is the most, yes, it is the most abundant, and it is the most therapeutically used target receptors. It is the G protein coupled receptors. So from its name, there is something we call a G protein, and this G protein is coupled with these receptors. Now, this is the lipid bilayer of a certain cell. Okay, here's the receptor, the G protein coupled receptor. A specific characteristic of this receptor is that it spins inside the cell 7 times. So this is 1 and 2 and 3 and 4, 5 and 6 and 7. Okay, the extracellular domain is the part where the ligand, the active site or the binding site of the ligand, while the intracellular domain is the one that is linked to the G protein. The G protein is 3 subunits. Okay, they are 3 subunits, and these 3 subunits are connected to each other. Okay, this is 1 and 2, and this is 3. Okay, these are called the alpha, the beta, and the gamma subunits. Okay, the alpha subunit is the one connected and the one communicating with the G protein coupled receptor. Okay, what is special about these receptors is that their effect is usually on a third different molecule, a third different protein or channel or enzyme or whatever. So here we usually have a third different molecule, okay, or protein, that the final effect, the ultimate effect, is going to be on it. Now, for example, this G protein coupled receptor is now connected to its ligand, so it is now stabilizing, this ligand is stabilizing its active state, so it is now active. Now, when it is active, it will come and activate the G protein. Activation of the G protein will make it separate, okay, so the alpha subunit, okay, the alpha subunit is now separated from the beta and the gamma subunits. Now this happens because this alpha subunit is now phosphorylated. Is it the alpha subunit itself that is phosphorylated? Actually, no. In this inactive state of the G protein coupled receptor, the alpha subunit is connected to a GDP molecule, okay, diphosphorus. So it is a GDP. When this G protein receptor coupled receptor is active, this GDP on the alpha subunit is replaced by a GTP. Okay, so now we have a GTP. Now this GTP will make this alpha subunit active and movable, and it will come to this part in here. For example, we have here an enzyme. Okay, so it will move. Let me make the drawing and I'll come back. So now this alpha subunit is connected to a GTP, which has replaced the GDP. It will be separated from the beta and the gamma subunits, and now it's gonna move and reach another molecule or substance. For example, the first example, this alpha subunit will go and activate a specific enzyme here, and a realistic example of this is the adenylyl cyclase. Or this subunit will go, okay, the alpha subunit will go to a certain ion channel and activated, and so it will be open, and this will move to the inside of the cell. Okay, now this adenylyl cyclase, when it is activated, okay, phosphorylated and activated here, this adenylyl cyclase will produce the cyclic AMP. And this cyclic AMP, it has the ability to go to certain proteins and phosphorylate them. So this cyclic AMP here is the second messenger. Okay, this is done through the adenylyl cyclase, through the alpha subunit, the activated subunit. It was activated by the G protein. The G protein was activated upon binding of the ligand. So this is the ligand activating the alpha subunit. The alpha subunit will be replaced from GDP with GTP. The GTP will go to a certain enzyme, activate it, give it phosphorus, and the adenylyl cyclase, this is an example, will go and produce the cyclic AMP. This is the second messenger. So you activated an enzyme through the alpha subunit that will go and produce the ultimate effect on the cell. Or for example, this alpha subunit will go to a certain ion channel and it will go and make the effect, like this work in here in the ligand-gated ion channels. Okay, هلا if we take the adenylyl cyclase as an example, you should know that the alpha subunit, when it goes to the adenylyl cyclase, and this is a very important example and the most abundant example, it will also activate what we call the phospholipase C. This phospholipase C will activate the IP3 and the DAG. Okay, when these two are activated, this IP3, always remember that IP3 is related to regulation of the calcium ions inside the cell. And remember that calcium ions are concerned with the contractility. So if this is the smooth muscle, for example, this activation will lead to the contraction by increasing the calcium ions concentration. And this drag will go to what we call the PKC and activate it. Okay, so remember this IP3 and remember this adenyl cyclase and cyclic AMP and the activation and phosphorylation of certain proteins. This is done by the G protein coupled receptor. I hope everything is clear now. One last thing that you should remember about these G protein coupled receptors is that they are included in neurotransmission, in olfaction, and in vision. Okay, and don't forget the second messenger thing in the G protein coupled receptor, and that's it. By now, you know what signal transduction means. When we have a signal, it is carried through the drug has the signal through the ligand. We go to the receptor and the receptor will take the signal and make certain changes, and these changes will be either direct on a certain molecule, a certain protein, or they will be through a second messenger, and so we will finally get a response or an effect in the cell. Now, everything in the body is under regulation, and so is the signal transduction. Now, the signal transduction is either under positive regulation or a negative regulation. That means that the body can do things to increase the signal transduction or do things to inhibit or decrease the signal transduction. Okay? Now, if this was a certain cell, this is the lipid bilayer, and these are the receptors on the surface of the cell. If this was a G protein coupled receptor, for example, okay, and here we have the alpha, the beta, and the gamma subunits. What you should know that upon binding of the ligand to this receptor and activating it, okay, this G protein coupled receptor will activate the G protein, and the G protein will go on and work on other things. Now, if this receptor is going to dissociate, because it never binds forever, okay, the binding is never forever, so here we have the dissociation of this ligand from this drug. The activity of the effect of the changes that are happening now because of this binding are now separate from the binding of the ligand to the receptor. يعني هاي مش مقرونة بهاي. يعني the alpha subunit will now go and work on its own regardless the binding of the ligand to the receptor. But this does not mean that it will be also active forever. It will finally be inhibited. But the duration of activation of alpha subunit upon binding and replacing of the GDP with GTP is different, okay, is longer than the duration of the binding of the ligand to the receptor in here. And this is what we call the amplification of the duration. Okay, and this is 1. Now, the other idea is if this was the heart, and the heart has beta receptors, as you already know, and these beta receptors are on the heart waiting for the norepinephrine to come and bind. Norepinephrine on the beta hearts will activate the sympathetic pathway. That it has a sympathetic activation, a sympathetic effect that will increase the contractility and will increase the pulse rate of the heart. Okay. Now, if these beta receptors are here and norepinephrine comes and binds to this heart, what they found not only in this is, I'm just taking this as an example, is that the ligand binding to these receptors here to make the effect, okay, is usually to a certain percentage of the receptors. So this means that in order to get the maximum contractility or the maximum effect of this ligand on these receptors here, we don't have to bind to all these receptors. So we always have these kind of free receptors that we can get the maximum effect without being used. Why is that? Let's assume that this heart here, the muscle of this heart is failing. This person is having a heart failure. Now, if we have the norepinephrine in order to increase the contractility, increase the rate to the maximum, okay, it has to bind to the beta receptors in here. Now, in the failing heart, the binding of the norepinephrine to the usual amount of the receptors will not give a sufficient and life-efficient contractility of this heart. Now, in the failing heart, these sparing or these sparing receptors are usually occupied, and we use them in the failing heart. So this is something good for the failing heart, that we have sparing receptors. So these sparing receptors are there, whether in the heart or in other parts of the body. They are there for the situations where we have abnormal things going, where we have weaker system, where we have failing heart. And another example is the cell with the insulin receptors in here. The insulin receptors are usually, which is very surprising, 99 of them are sparing receptors. And the other 1% is what is usually used in the normal cell, that is to make glucose get to the inside of the cell. While in the heart, the sparing are 5 to 10% of the total. Okay, so now we have two ideas: the amplification of the duration and the sparing receptors. Now, the third thing is the amplification of the magnitude of the signal itself, away from the amplification of the duration. Now, the amplification of the magnitude of the signal, we can explain it if we come back to the G protein coupled receptors. Now, they say that the alpha subunit here, the activated alpha subunit of the G protein with the which is bound to the ligand, this can, as we said, it should go to a certain enzyme or an ion channel and activate it. They found that the activated part here can go to multiple enzymes, to multiple proteins, to multiple ion channels and activate them. That is, if this G protein coupled receptor is going to work on a certain ion channel and activated it and open it. Now, this is number one. It will go and get back to another ion channel. This alpha subunit will separate from this, go to another ion channel and activate it, and then go to a third ion channel and work on it. This is done by only by being activated by only one ligand. Okay, this is the first thing. And the other thing is that this enzyme, okay, if we are working, if it has activated an enzyme, this enzyme is going to activate multiple adenylyl cyclase. It's going to activate multiple second messengers. This adenylyl cyclase here, it's going to produce multiple second messengers, multiple cyclic AMPs, and these will work on many proteins and many other stuff after that. So the response will be amplificated. It will be much bigger than the first signal, which is the single ligand here. Okay, so the second thing we have here is the amplification of magnitude. Okay, this is the magnification, the amplification of the duration. This is the amplification of the magnitude, and this is the sparing, which is the third one. Okay, now the second idea is to regulate, to downregulate, or to make the signal transduction less. This is done, or this has two main concepts. Okay, the first concept is what we call, or without mentioning what it is called, let's talk about the okay, now if we take this as the response against time, okay, and here is the cell, here's the receptor, and we brought the ligand here, okay. The ligands here are bound and produced the maximum effect in the cell, okay. We brought this G protein coupled receptor, it produced a certain effect, the cyclic AMPs, the calcium concentration, whatsoever. So here we got the maximum response, okay. Now, they found that if you brought the ligand for the second time here over a short duration, okay, you will get a response, but this response will be less than the first response, okay. Compare this one to this one. But if we come here and wait for a certain period of time, longer period of time, and we brought the ligand here, okay, this is going to produce the same effect that we produced for the first time. This means that if ligand 2 was produced on a duration longer than this, yani if ligand 2 was here, okay, it will produce the maximum response. Why? Because here we have something called the refractory period. What is the refractory period? Let's simplify this, okay. This is the ligand 1 here, okay, this is ligand 1, and this is when we introduced it, we got this response. Now after the first response, you should wait for a certain time, okay. This is called the refractory period. If you introduce the ligand, okay, to the receptor and during this refractory period, you are not going to get a response similar to the maximum, or you are not going to get the maximum response here. Why? Because the drug is still resting. It is still tired from this first response. So now, when in response number 2, we waited outside the refractory period, let's assume this is the refractory period of the ligand, of the second response that we get, we are now outside the refractory period. We introduced the ligand for the third time here, so we are back to the maximum response. So in this ligand, okay, in this ligand introduction here, if we had the response, okay, here, and we introduced the ligand for the second time anywhere during the refractory period, we're going to get this or this or this, but you are never going to reach the maximum. But if you introduce it outside the refractory period, you will get the maximum effect back again, okay? And this is what we call the desensitization. Desensitization is when the desensitization desensitization. It is when the receptor is tired. It is still tired and it cannot give you the maximum response. Okay, its usual maximum response. It's tired from the first response that it elicted, so you should wait until it gets its power again and will give you the maximum response again. Now the other thing is when you have the cell in here and this is the receptor here. Okay, this is the receptor. We have, for example, 2 receptors here. Now the receptor is there in order to receive a signal. This signal will come and produce a certain effect and leave. This is how the cell understands the idea of the receptor. Okay, this receptor or this cell will only accept the activation of the receptor for a short period of time and not over repeated periods. Okay, and this is why we have the desensitization concept, because the cell will protect itself, because this is not what it produced its receptor for. It did not produce its receptor in order for them to keep activating it, okay, to keep it awake, to keep it working. So this is why we have the desensitization. The other concept is the concept of down regulation. What do we mean by down regulation? If this is the cell and here we have the ligands, okay, the ligand here came and bound to this receptor, okay. As we said before, that this ligand should dissociate from the cell. If this receptor bound to this drug for a very long period, to this receptor for a very long period of time as a continuous binding, okay, this will keep the cell working. It will keep producing an effect in this cell. Now the cell will understand that there is something wrong. There's something keeping it working and keep it activating. This cell cannot dissociate this ligand from the receptor. What it will do actually is that it will get this receptor to the inside of the cell, okay. It will get this cell to the inside, it will get this receptor to the inside of the cell. It will endocytose it, so by endocytosis. Now when we go and endocytose this receptor, the ligand will come and it cannot cross the cell, so it will come to the cell surface, no receptor, so no effect. Now after it endocytoses it, here we have the endosome. For example, this now is here, this is the endosome, and the endosome here is having the receptor with the ligand connected to it. It has no effect. The cell will either degrade it and get rid of it, so it has less number of total receptors of this certain family of receptors, or it will keep it in the endosome frozen until the signal is away, and it will recycle it and put it back on the cell surface. Okay, so we have 2 options, and this is what we call the down regulation. Again, to repeat it, down regulation is when we have continuous okay, in comparison with desensitization is when we have repeated activation. Down regulation makes the cell endocytose the receptor, okay, and after endocytosis it will either recycle it, so it will be used again later on and put on the cell surface, or degrade it, okay. While the desensitization, it will make the cell unresponsive during this period of time. Not totally unresponsive, but it will make it unresponsive to the to reach the maximum response, Okay, this is the downregulation, and this is the desensitization. I hope that you got the two ideas and you can compare with them easily.